as blastomeres. So zygote will first change into 2 cell situation of blastomeres, then 3, then 4, and finally morula, and then blastocyst. Now what is blastomere? Blast means to form and meres means the monomer. So it is a fact that if you take any of these 4 cells out and you start growing it into a separate medium, it can give rise to a complete new individual of identical type. Reason, these are stem cells. Stem cells are those cells which have the total potential to give rise to a complete individual. So one cell retains the capability to give rise to a new individual. This is how twins are born. So now what happened next is, this blastocyst is going to be the stage which will undergo implantation. Please remember that. And at the blastocyst stage only, when the right place of implantation has come, zona pellucida will start breaking. Now, what is the role of zona pellucida throughout this process? Zona pellucida does not allow the implantation to happen at the wrong place, because sometimes what happens, as the structure is moving down through the fallopian tube, it may get implanted at the wrong place, maybe somewhere in the isthmus, maybe somewhere in the wrong place, or in the fundus portion of the uterus. So to stop the implantation to happen at the wrong place, zona pellucida persists. Reason, the cells which are just under the zona pellucida are phagocytic in nature. Phagocytic means they are releasing enzyme and they can cause the breakdown of the surrounding medium, and this can cause the blastocyst to undergo implantation at the wrong place. So when the right place of implantation has come, then only the zona pellucida starts breaking, and it breaks into pieces. And now what will happen? Blastocyst is exposed. What is blastocyst? Now let's understand. Morula consists of all similar type of cells, but the blastocyst will have 2 types of cells. The outermost layer of the cells is called as the trophoblast. Tropho means food, blast means providing or forming. So the cells which will provide nutrition, it's not actually forming, they are providing, because they are imbibing, they are absorbing from the surrounding, and then they are passing it on to the central portion. The central portion is a cavity. So this cavity is called as blastocele, and this cavity is full of nutritive liquid. And at one corner you can see I have drawn some cells. This is the inner cell mass, which will later give rise to embryo or the next generation. Now this structure is round, so scientists have given it names. The end which side the embryo is present is called as the animal pole, or is also called as the embryonic pole or embryonal pole, and the pole opposite to it is called as the abembryonic pole or abembryonal pole. So 2 ends are there. The end which carries the inner cell mass will later give rise to the next generation or the embryo. So I hope this much is clear to you. Now by this time 6th or 7th day has come, and blastocyst has already reached the uterus, so it will undergo implantation. Now by the 8th day, the trophoblast develop 2 layers. Now what are the 2 layers? So now please pay attention here. Imagine this is the 7th day. Trophoblast has come in contact with the endometrium. This is the line which I have drawn as the uterus or endometrium. Now this trophoblast starts developing into 2 layers. So the inner layer will continue to exist as a cellular layer, but the outer layer will become syncytial. Remember 11th class syllabus? Syncytial means multinucleate cells in the case of animal cells. So when animal cells are multinucleate, they are called as syncytial. So the names I have clearly mentioned here. The inner layer of the trophoblast is called as cytotrophoblast, and the outer layer which is syncytial in nature is called as syncytiotrophoblast. Now this layer is very much important because it will develop the chorionic villi or finger-like projections, which will develop into placenta later on. Now what exactly happened? By this time the inner cell mass will differentiate into 2 layers. Now you can see I have drawn the same thing into 2 layers. So the outermost layer which you see there are called as the hypoblast, and the other layer is called as the epiblast. Hypoblast is below, epiblast is above, because in books they will show this diagram in this manner. For convenience, because I learned always in this manner, I have drawn like this. So hypoblast, hypo means below, epi means above, you can easily make out. So hypoblast layer is made up of cuboidal cells and epiblast cells are columnar in shape. Now what happened after this is, there starts development. Okay, now please remember there is a trophoblast present here. So now a gap starts developing between the epiblast and the trophoblast.